So in our group, we are interested in enzyme design. We are typically combining computational design and directed evolution to create novel enzymes. In most of our projects, we start by designing some active sites, so either by putting in a new active site into an inactive protein scaffold, or nowadays actually using diffusion to build new proteins around active site models. But then after experimental validation, we move forward and usually have to improve the outcoming enzymes by directed evolution in the laboratory in rounds of mutagenesis and screening. And in today's talk, I'd like to, in the first half of the talk, tell you a little bit more about the science that we have been doing and the ideas that have led up to developing AI.zymes. And in the second half, I'm going to walk you through how you can actually run AI.zymes on your own design projects. So creating enzymes by design and evolution. Historically, we're actually coming from the experimental side. One of the first projects that we have been actually working on was taking designer enzymes that have been made by collaboration partners and enhancing their activity by evolution. And one of the reactions that we are very interested in the group is the Kemp elimination. The reaction is depicted over here. It's a very simple one-step reaction. All you need is a catalytic base that deprotonates the substrate, and you right away go to the product. And although the reaction is relatively simple, it's a very nice benchmark for our ability to design new enzymes. Because from a theory point of view, we know quite well what we need to do. We need to position this catalytic base in a very precise geometry relative to the substrate to promote this deprotonation reaction. There's no issues with selectivities, side reactivities, etc. One of the first projects that we've been working on actually was to take a designer enzyme that has been designed by Steve Mayo, and although the enzyme was active, the activity was unfortunately quite low. So we subjected this enzyme to directed evolution, and over 9 rounds of mutagenesis and screening, we were able to enhance the activity by 3 orders of magnitude, now really leading to a catalyst that starts to look like an enzyme that you could find in nature. And so having done this work, we really started to wonder, is there maybe something more that we can get out of these evolutionary experiments? Because in the end of the day, nobody needs another Kemp eliminase. So maybe there's something that direct evolution can teach us about how to improve computational design. Maybe if we understand what the mutations are doing that we introduce during evolution, we can derive design rules that will help us to make better catalysts in the first place. And this is an idea that we have spent quite a lot of time on, really dissecting the effects of individual mutations, looking into how the electrostatics and conformational dynamics of the protein are being modified by evolution in order to enhance activity. And to cut a long story short, what we and others have seen in countless evolutionary trajectories is that particularly electrostatics and dynamics are 2 factors where we really need to improve in enzyme design to make better catalysts. So the next big question would be, now that we have learned these lessons, how can we integrate them into enzyme design? How can we tell a design engine such as Rosetta or ProteinMPNN to design, for instance, for a certain electrostatic environment at the active site? And this is pretty much how the idea was born to engineer AI.zymes. AI.zymes is a very modular platform in which we're trying to integrate a range of different protein engineering algorithms. And the fundamental idea of AI.zymes is that we would like to do evolutionary design. So we run rounds of computational design and then selection for variants that have the catalytic properties that we think are important, beneficial for catalysis. And so one fundamental hypothesis that underlies AI.zymes is that we first do some sort of neutral, quote unquote, design step where we run these protein design algorithms to enhance the stability of our enzyme substrate complex. And then later from these stable structures, use evolution and use selection to enrich these catalytic features that we think are important for catalysis. Another beautiful thing in AI.zymes is because we're running rounds of design and selection, during every design round, we can use a different design algorithm. So sometimes AI.zymes uses Rosetta to redesign the enzyme. Sometimes it uses an AI-based tool such as ProteinMPNN. Likewise, on the selection side, we can now start to throw all of these beautiful protein analysis tools that are out there onto our system in order to enrich for the property we're interested in. So in the original publication, we were using ESMFold for structure prediction, and then paired this with FieldTools, which is a computational algorithm that we have written to assess electrostatic features at the active site, so these properties that we would like to enhance. Increasingly, we're also interested in combining this with molecular dynamics simulations. Admittedly, we are at the moment only doing this after the AI.zymes design run, but we're working really hard to actually integrate MD simulations into the selection step so that during every design cycle, we run a short MD simulation on the protein that we have made in order to select enzymes that have the catalytic and dynamic properties that we are interested in. AI.zymes, to cut a long story short, the motivation here is that we, in a very modular fashion, want to combine the best of all worlds, want to combine the advantages and strengths of all of these algorithms that we're using, both for established but also emerging design technologies. So once we have written AI.zymes, we decided to benchmark the algorithm on Kemp eliminases once again. This time we're using targeting the Kemp eliminase activity of a natural enzyme. So ketosteroid isomerase, KSI, has promiscuous activity for this reaction, and we chose to try to enhance that activity by design. At that point, ligand MPNN wasn't yet available, so we decided to split the protein in two, treat the active site with Rosetta that can see chemical ligands, and then treat the rest of the protein scaffold with ProteinMPNN. So in a way, we're really using every algorithm to what we believe is the best, really exploiting the strengths and weaknesses of each of these algorithms. What did we try to optimize? Of course, we're still after protein stability. So we have the stability score that we're trying to increase, which reflects the total score, the total Rosetta score of our enzyme substrate complex. But then in addition to this, we have some more enzyme-specific features that we're trying to improve. So we are calculating an interface score, which represents the interaction energy or the interaction score of our substrate or transition state model with the enzyme active site. And then we even go one level deeper and also score for the catalytic geometry in a score that we call catalytic score, which really assesses how well are the catalytic residues aligned with the substrate according to what we know from DFT calculations. Last but not least, we're also looking into electrostatics here using our FieldTools script. And so electrostatics can actually be electrostatic interactions can very nicely be quantified using electric fields. And this is what we get from FieldTools. And to very simply explain the idea here is in the Kemp elimination, this is a deprotonation reaction. We need to move this proton, this positively charged particle on the slide from the substrate up to the catalytic base. And in a very hand-wavy way, it's probably going to be beneficial if we put a lot of negative charges on one side of the protein and a lot of negative charges on the other one. This is a strong simplification of how electrostatic catalysis is actually working, but I guess this is helping you to get an idea of what we're after when we're looking into electric fields and electrostatics. How did AI.zymes perform? So we first started to benchmark the algorithm purely in silico. So we compare a non-evolutionary design run where we always start with the same input structure to an AI.zymes run where we do evolutionary iterative design. And here you can see the fitness landscape of the non-evolutionary design run where I'm plotting the predicted transition state affinity, so the affinity score. And you can see that we're generating a couple of variants that actually have higher activity than where we started from, which is somewhere in the back over here. However, once we do evolutionary design, we can really see the strength of AI.zymes in gradually traversing the fitness landscape and enhancing fitness. So not only are we exploring a much larger range of the fitness landscape, but we're also getting much higher values in this case for the predicted transition state affinity. I mentioned very briefly that we are looking into electrostatic catalysis. So how did we do in terms of designing the electric fields? One thing that is really important to note here is that the design algorithms that we use, Rosetta and ProteinMPNN, they did not target the electric fields. They were purely aimed at maximizing the stability of the enzyme substrate complex. Moreover, something that I'd like to note here is that designing electrostatics is actually quite challenging because usually you need to sum up a lot of very small effects all over the protein scaffolds that add up to an electric field at the active site. And also you need to be very careful not to introduce any charged residues in your hydrophobic core, which will be detrimental to the protein stability. And here we see one of the strengths of AI.zymes and the approach that we're choosing, because the design algorithms only care about protein stability. We're only selecting for favorable electrostatics in a pool of stable proteins. How did we do? This is a plot of the evolutionary history where we are plotting the calculated electric field at the active site over the design generations, and you can see that we are able to gradually improve this property, at least in silico. Now, when you do enzyme design, it's absolutely critical that you also always experimentally validate your designs, and this is also what we did here. So we tested 7 designs in the lab. Interestingly, we mutated almost half of all of the amino acids in the protein. So this is a 120 amino acid enzyme, and we have 60 mutations on average in the tested designs. Nevertheless, All tested designs were monomeric, all were thermostable, and all were really well behaved. And the best one was actually also improved in activity by almost 8-fold. So this is what we have in the paper. During the second half of my talk, I'd like to give you a little bit more of a walkthrough of how we use AI.zymes and how you could actually use it in your design campaigns. So installing AI.zymes is actually quite simple. You just do pip install AI.zymes. I would nevertheless recommend that if you're interested in giving this algorithm a try, that you reach out to us and get access to the development version for AI.zymes. We're more than happy to provide you access. Just send me an email. The advantage here is that we have really substantially expanded the design capabilities compared to what we have in the paper from summer last year. So what are these expansions that we have in AI.zymes? So here you can see an overview of all the modules that we have in the original paper. In terms of design modules, we're now using soluble MPNN, which is slightly better than ProteinMPNN. And we also added a null module, which is actually not doing any design, but it's really helpful if you just want to score a range of variants and you're just interested in how do they behave after structure prediction and relaxation. On the scoring side, we actually substantially expanded our capabilities to assess the catalysts that we have designed. So we're now also offering to use co-folding models where you can predict protein ligand complexes. So we have Boltz2, AlphaFold3, Chi1. In addition, we also established SEEX2 to quickly provide multiple sequence alignment for the structure prediction. We also started to integrate Ember into AI.zymes. This is at the moment only doing structural minimization. However, we already see that doing structure minimization in addition to Rosetta relaxation is actually really beneficial to get more accurate structures. And not only is it more beneficial to get more accurate structures, we can also use then the molecular dynamics engine and the force fields to estimate interaction energies. For instance, using GBSA. Again, this is not running MD, this is just scoring the static structure, but we can use these really high-level force field tools that we extract from that have been developed for Ember. In addition, we also established tools to optimize redox potentials, particularly for heme cofactors, and also to score protein interactions better. Altogether, in the original paper, we were with AI.zymes, with the original version of AI.zymes, you can only design substrate bound to a protein. What we now can do in AI.zymes is we can have multiple protein chains, we can have multiple substrates, we can also have no substrates if you would like to design protein interactions. We can have covalent intermediates as well as non-natural amino acids and define the protonation states of some of the side chains, which might be important for certain catalytic mechanisms. So for the rest of the instructions, I will focus on AI.zymes 2.0. If you want to use AI.zymes, there's a working example on the GitHub. It's going to be very similar to what I'm showing you right now, but there might be some differences. So if you opt for the older version, I would recommend you follow the example notebooks that we provide. One thing that we now also added to AI.zymes 2.0 is that we provide another set of functions to do automated ligand parameterization. I don't have time to go through this, but the idea here is that you can run rounds of structure prediction with bolts of only the small molecule ligand, QM minimization with ORCA, so you get really accurate structures, and then parameterization both for Ember and Rosetta. Because in AI.zymes 2.0, we're now using both Ember and Rosetta, we need to provide parameter files for both of these algorithms. So what you will typically get is the params file for Rosetta, but also a prep.i and FRC mod file that Ember needs. We're also getting a mol2 file that is optionally to be provided in AI.zymes, but we use this to unify the atom naming scheme, which is very helpful to prevent any issues further down the line when you start to combine different computational algorithms. Now, how do you run AI.zymes? The first step is to make an instance of the AI.zymes main class. Here you only need to give the home folder in which you want to work. So this is your working directory. When you do this, will automatically create an input folder, and you will have to put all of the input structures, that input files that AI.zymes is looking for into that input folder. Once you have this, we need to run the setup, and this is really the heart of setting up AI.zymes here. We're fixing all of the settings that we need for the future design. Then this is also the only point where you actually interact with the design algorithm itself. So we're setting up all of the design settings, and then the idea is afterwards, AI.zymes will just run on its own and do the evolutionary design all by itself. To briefly walk you through some of the settings that we have, so there's some job-specific settings where we define how many GPUs and CPUs we would like to use. AI.zymes is coded in a way that we're trying to maximize the resources that we provide, and we typically give the number of GPUs and CPUs that we have on one full GPU node, but these numbers obviously can change depending on your hardware. We also need to define how many designs we do based on the parent structure. So here the idea is, because AI.zymes is an evolutionary design algorithm, it needs to do selection based on a pool of variants. And we first need to generate this pool by performing design based on the parent input structure. And here we define how many designs we do for this. There we go. The next point are some more system-specific settings. So you define the name of your protein, or you define your ligands. So because we're now doing ligand-protein co-prediction, you need to define how your ligand is defined. For instance, we can do this via SMILES codes or CCD codes, and then give the definition itself. So here, we're having one ligand that is defined by this SMILES code over here. Lastly, we need to adjust the design settings. And here we first have a list of design models that we use for the parent design. So these are design steps that will be run on your initial input structure. And then once you have made enough design steps as defined previously, the AI.zymes algorithm, the evolutionary design algorithm kicks in, where we can actually sample between different lists or different sets of modules that you can define however you'd like to. And here we're also now giving a probability. So with a 50% probability, this would run Rosetta design with everything that comes afterwards. And with a 50% probability, we would run soluble MPNN. Down here, we're also defining the residues that we allow Rosetta to redesign. And by default, ProteinMPNN or soluble MPNN in this case will target all of the other residues that we haven't defined here. But you can also set this manually yourself. You also need to define what scores you would like to optimize on. So here you can see a list of scores that we've chosen for this specific run. And for the catalytic score, which was the catalytic interaction between the substrate and catalytic residue, you furthermore need to define the name of your Rosetta NSTES constraint file, as well as the two residues between which you actually want to define this constraint. Once everything is set up, you run the setup, and if everything is all right, you get this nice message, happy AI.zyming. If not, we try to make AI.zymes more and more talkative, so it will let you know in case some files are missing or something is wrong. After the setup, you need to submit your AI.zymes run. So submit controller will just submit a job to your HPC queue. AI.zymes is quite smart, so you cannot have two times the same job in parallel. However, something I should note here is that once one job has finished, for instance, you timed out, you can always resubmit the job, and then AI.zymes will continue designing based on where it left off before, for instance, you reach the wall time limit of your HPC queue. Great. So now we're done with the design. How do we identify our improved variants? How do we find better designs? So the first step will be to just do some statistics. So there's one function that will tell you how many designs you've made. There's also some other quality of life options here where you can get some additional information, but this will essentially tell you how many designs you have been running, where you're at, what is currently happening. It will also print the log file if you were interested, if there were any error messages. The next function to use is plot, where we are plotting the design results. And here I'm just going to very quickly walk you through the various plots that we're providing. So first of all, you get this evolutionary tree. I've already shown this before. So here I'm projecting the final score, which is essentially an aggregation of all of the scores we're working with in AI.zymes. And you can also see, for instance, here, these are individual designs that actually failed. Maybe the sequence was so bad that the structure prediction didn't work. We don't know why they failed, so they will just be ignored, and AI.zymes will continue not actually touching these variants anymore. In addition to this tree, we actually get a whole lot more of very helpful plots of the various variants that we have been generating, either histograms, plots of the scores against the design index, or plots of the scores against the design generation. So here we have quite a few generations, which is why this plot is a little bit busy. But what we'd like to see is that your scores are actually starting to converge, that they plateau, which is a very good indication that the design is actually finished, and you can start to now extract the best variants for characterization. How do we extract this? For this, we also supply one function, best structures, which will try to identify the best variants in all of these scores. So you give like a cutoff for the final score, which is, again, like this aggregation of all of the scores, which is making sure you're trying to select the best scores and the best metrics of your whole AI.zymes run. We're also showing your parent designed variants. Here, we didn't do any design, so this is really the parent variant, the original sequence, so you get a good estimate of how you actually also improved during the design run. Not only do you get these nice plots, this function will also create a new folder in which it will save all of the structures that you've been working with. All of the hits will be saved to this folder over here, and here's a snapshot of the folder itself, so you get all of the structures. But in addition to this, you also get this .pml file. This PyMol script will automatically load all of the structures that you have and change the visualization in a way that you can really nicely see what has been happening during AI.zymes. To give you one example, so I ran the script for the files I showed you before. And as you can see here, the residues are color-coded in different ways. And so essentially, in this specific design run, all of the green residues were residues that have been addressed but targeted with Rosetta design. All of the yellow residues are residues that have been targeted with MPNN. And all of the salmon residues are residues that were actually fixed that we didn't allow to mutate during the design run. So you can also specify this. And what is quite nice, once you export, once you get the best structures, you run this function, you can actually really also visualize which residues you targeted to double check if all of the selections actually did what you wanted them to do. Great. With this, I'm already at the end of the presentation. To wrap this up, what we are really interested in our group is to expand the space of designable features, to go beyond stable proteins, maximizing protein stability, but really think about other properties that we would like to design, especially to achieve high catalytic efficiency. I've talked today a little bit about how AI.zymes 2.0 can now handle multiple substrate, oligomeric proteins, do protein ligand co-folding. We in the group are very interested in also designing more redox active proteins. So looking into redox potential, photocatalysis, and electrostatic catalysis is something that we are actively working with, in addition to now delving into de novo enzyme design and trying to combine AI.zymes with these other de novo design engines. Also, although this is not at the heart of where we want to go with the group, AI.zymes can be used for binder design, so you actually don't have to have a ligand around in your design projects. We can do covalent intermediates. We're now going more into also looking into substrate selectivities and trying to establish better methods for protein dynamics, multi-state design, and also data-driven design is something that we are very interested in. So I'm highlighting all of these things, particularly because some of these lower points in this list are very experimental features that we're currently working on in the group. So AI.zymes is actively developed. We have probably many more options than what I could show you today in the presentation of how you can use AI.zymes. So please do reach out if you have any questions for your specific design challenges. We're also more than happy to help you set up AI.zymes for your design problem, and maybe even think about how we can expand AI.zymes with additional modules. Because as you've seen, since we've published AI.zymes in summer last year, the number of available modules and design capabilities has really exploded, and we're really eager in integrating more and more functionalities into AI.zymes. With that, I'm at the end of my talk. I'd like to thank all of my group, both current and former group members. I highlighted the people who were actually involved in this work in bold over here. And with that, I'd also like to thank you for your attention. And I'm very much looking forward for any questions that you might have.
Great. Thank you very much. It was an amazing talk and an amazing library. If you have questions, paste them in the chat. If you want to ask them yourself, raise your hand. But I'll warm you up with a little bit of questions. So can you, since we're talking about protein design pipelines, can you go into a little bit more details about how you integrate the tools? Are you using a Docker container behind the scenes? Are you calling them from the command line? How does that work?
I see. So essentially, AI.zymes is pretty much a glorified Python wrapper that will call like all of the executables individually. So you need to have all of the tools that we're using installed. So you need to have Rosetta installed. We're currently very much converging on using Boltz 2 for structure prediction, which luckily is much easier to install than some of the other design structure prediction algorithms out there. But yeah, so at the moment, you need to have all of the individual tools installed. We are very keen on, yeah.
And then you basically write your input files, run the thing, and then parse the output, right?
Exactly. But so what AI.zymes is doing under the hood, it's like collecting all of the information from the various design structures, selecting which variant to then redesign, also deciding what design method to use, and taking care of the job scheduling. So we are really aiming at maximizing the research usage. So when we are defining, for instance, in our system to use 72 CPUs and 4 GPUs, and it's really maxing out the available resources by always spawning new calculations.
Can you comment on your fitness function? Is it just a linear combination of the...
Great point. Yes, absolutely. So at the moment, it is just a linear combination of whatever you'd like to score on. So you can actually manually define this in this list over here. You can also add weight for each of these parameters. So if you want to like balance this by any values that you might think are important, that is possible. We're currently actually working on a more data-driven approach where we're trying to integrate feedback from experiment to really try and figure out which of these scores that we're calculating are actually most critical for catalytic activity. And I guess maybe as a side note, I feel like this is not pretty much where the field is going to be heading with all of these beautiful tools that are coming out now. Our design abilities are getting better and better, and we need to start asking these questions. Okay, what do we actually need to design? What are the fitness features that we need to enhance in our case to generate high catalytic activity?
Oh, very cool. And I guess just another less technical question. You're basically tied to a Slurm schedule, right? Before people start asking.
Okay, yes, exactly. So at the moment, all of this is running with Slurm. We are very motivated to also get this to run with other HPC systems because we here only have access to Slurm. This is what we have, but in case there's someone willing to develop and troubleshoot this with us, we are more than happy to give this a go.
Great. Now we'll start with user questions. So one question is, can we integrate HEDOC or other docking instead of AlphaFold for co-folding? And I'll generalize this a little bit. So how extendable is your Python code behind the scenes?
Very much. So this is something which I was trying to illustrate here. So all of these additional modules were super, super easy to add to AI.zymes because all of these modules pretty much need to. So all of these modules are built in a way that they either take a PDB or sequence file as input and will put out a PDB or sequence file. We can right away combine this with whatever algorithm we're interested in. And this has been very straightforward to expand the capabilities, which is also something that was really important to us developing a design tool in this very fast-paced times that we're in.
Very cool. So what does the efficiency look like for the entire pipeline? I think the question here is, how many designs would you recommend testing experimentally? Do you do like 24, 96, higher than that?
Maybe to go first one step back in terms of like how many designs do we actually need to make? We typically see that AI.zymes plateaus after 10,000-ish designs. And we think that this is because we hope that this is because we're sampling the sequence landscape a little bit more efficiently, actually restricting or biasing against putting too many mutations in Rosetta or soluble MPNN. So we're only doing like very small steps along the sequence landscape. Similarly, probably to directed evolution, we're doing many more mutations than what you would do in evolution, directed evolution. But the idea is to gradually walk along the fitness landscape. So what this means is what we've seen is we need to have probably an order of magnitude less designs than what we would need to get a comparable efficiency by just, let's say, just running Rosetta in a standard way. Then when it comes to experimental testing, we are now really pushing towards always testing 100 variants for each of our design projects. The reason isn't that much because of the success rate. It's much more about getting the statistics right, because when you only test a handful of variants, you never really know where you're just lucky or is the algorithm really as good. However, once you start to get these numbers, once you start to get the 100-ish designs, I do believe that you can really get a good feeling of the distribution of values that your design pipeline can actually achieve.
Very cool. And this is a very simple question. You're using transition state geometries for the ligand, right? So this is what gives you the enzyme activity. Okay. Can AI enzymes model direct protein-metal interactions without ligand?
Yes and no. One thing to think about AI.zymes, whatever the algorithms that you're working with can do, AI.zymes probably can also do, hopefully, if we tune it right a little bit better by combining these different methods. But whatever, let's say, I don't know, bolt structure prediction can do, and then you can minimize, I don't know, in Ember. If that works individually, it will also work in AI.zymes. So thinking about metals are always like quite difficult in computational workflows. But if, for instance, you have like very good parameters for your metal, for your force field so that it works in Ember, and you can decently accurately predict the binding geometry using bolts. So this should also work quite well here. I should probably also note that we have a couple of ways how to like bias all of these steps within AI.zymes. I didn't really have time to go into this, but there's much more that we actually enable. So for instance, in bolts already offers like a way to enforce bonds or contacts or define binding pockets. And this is also something that we allow in AI.zymes. So when you do structure prediction with bolts, you can, for instance, say, okay, this is the binding pocket that I would like to target to make sure that, especially in the beginning when your enzyme isn't as good, your prediction is really biased towards this region where you would like to have your substrate or your metal.
How do you do that technically? Do you just paste that in your the big configuration file at the start?
Exactly. So everything is controlled in the setup file. So there are a couple of flags how we can actually control the behavior of bolts and tell it to introduce these bonds, define the pocket. So the idea is really that once you did the setup, it's completely hands off. You just need to resubmit and wait until you have the designs that you like to have.
And how hard would it be to enable other workflows? Can you do binder design? Can you do motif scaffolding? Can you do symmetry?
We're actually currently working on symmetry. In terms of binder design, yes, we can do this. So we actually managed to completely eliminate the need to have a substrate in there. So this is quite hard-coded in the original version of AI.zymes. Here, we actually don't need to define a ligand anymore. And then it will try to design chain A in your protein to maximize the interaction of chain A with the rest. Yes.
Where do you define the size of your protein? There's also a question of how big proteins usually work well on this scale. Does 200 amino acids work, or bigger or smaller?
maybe I realized there's one part of your question that I ignored, which is, can we do motive scaffolding, etc. There, one point really importantly, so AI.zymes is intrinsically an optimizer that runs on a full-length model. So you need to have your full protein scaffold with your active site model in there. But so our idea here is, again, with AI.zymes, we are combining established tools. We actually want to rely on other tools such as RiffDiff, such as RFDiffusion, to generate these input structures that we can then further refine in AI.zymes. So we're currently actually working on showing that the designs we get out here are better than what we get out by just doing RiffDiff. So this is currently work in progress, but just looking at the structures that we're working with, it actually looks quite promising that this is really adding something to what RiffDiff is doing. Now, talking about size, this all boils down to like the performance of the methods that we're using. We are typically, so for the Kemp eliminase de novo design case here, we're using 200 amino acid proteins, and then like within a couple of days, like maybe a week on a single GPU node, we can complete the designs. We can get to 10,000 designs easily. Obviously, the bigger the protein it becomes, the slower it gets. But again, here the performance is really limited by the underlying design engine. So if you know how fast a Rosetta works with your input protein and how fast you can predict the structure of your protein with bolts, this will determine how quickly you can push this through AI.zymes.
Perfect. And like for binder design, then you would also have to give it like an initial binder, right? And then optimize that.
Exactly. So intrinsically, AI.zymes is an optimizer of input structures. And this is also the philosophy that we're having in developing this. So we want to integrate established and emerging technologies, tools in one coherent workflow to make these design tools interact better with one another than what they could do on their own.
This is again a technical question. How do you pass information from one workflow step to the next? Because you also always have some workflow specific things. Do you have a general pose or something?
Hang on, let me find I think I had this on one of the slides. Let's try and find this. Yes, essentially we are building this CSV file, which essentially keeps tracks of all of the designs. So per design, there's one line where we just collect all of the information that are generated as we go along with the design. And in here, we also keep track of what are the next modules AI.zymes is supposed to do. So this is defined whenever you start a new design index, so to say, a new variant. And then what AI.zymes does is just working through this. And essentially, this allscores.csv file has a laundry list of different modules to run on different indices, and it will just try to work them off as efficiently as possible.
Perfect. And this is also enables you to restart.
It is able to restart, exactly. Very importantly, you can run submit controller as often as you want. Like in case it crashes or you time out, you just run this command again, and it will seamlessly continue running.
Perfect. And I think last question, because we're running out of time, is it possible to contribute to this repository, or more generally, how are you open to PR, pull request? Times 2 is not out yet, so.
Exactly. So it is not out yet, but if you have a project that you're interested on using AI.zymes 2 for, please do feel free to reach out. We actually already established a whole bunch of collaborations with people who were interested in using AI.zymes, and actually with every use case that we have to challenge our design pipeline on, the whole program is becoming more and more robust and versatile, and we're introducing more functionalities. We are very happy to collaborate and to try and either help you tailor AI.zymes 2.0 to your design challenges if it's not yet possible, or co-develop. Absolutely.
Perfect. Thank you very much again. I think there's a few more questions in the chat if you want to go through them later.